Hi, today we will be studying about chromatin structure in molecular biochemistry. As we know that if we stretch a DNA from cell, it would be around meters long. So how DNA is fit into the cell? It is wrapped around protein called histone proteins. So DNA is wrapped around histone proteins to fit into the cell, as cells are about micron meters in length. So there are four types of histone proteins: H2A, H2B, H3, H4. As we know that DNA has negative charge on it, and histone proteins have positive charge. Histone proteins have positive charge because of two types of amino acid, lysine and arginine. Those amino acids are, one is lysine and another is arginine. Okay, one is lysine and another is arginine. So we will discuss about main points. Point one is, during which phase of the cell cycle does a replication of DNA and synthesis of histone occur? The answer is S phase of interphase. Our other point is high yield topics. What are the structural differences between mitochondrial and nuclear DNA? What are structural differences of mitochondrial and nuclear DNA? Mitochondrial DNA is circular, not linear, and does not utilize histones. And no histones. How does histone deacetylation decrease DNA transcription? How does histone deacetylation decrease DNA transcription? Removes acetyl groups, tightened coiling of DNA, decrease transcription. Our fourth point is, what are the components of nucleosome? So components of nucleosomes are two loops of DNA around histone octamer. Histones are H2A, H2B, H3 and H4. Which two positively charged amino acids in histone are primarily responsible for binding negatively charged DNA during chromatin formation? So those amino acids are lysine and arginine. Lysine and arginine. Lysine and arginine. Which epigenetic modification of histone has been implicated in Huntington disease? Histone deacetylation. Which epigenetic change silences one parental gene copy through genetic imprinting? DNA methylation. Methylation makes DNA mute. DNA methylation. Methylation makes DNA mute. Another is what effect does methylation of CpG islands within the promoter region of gene have on transcription? Decreases gene transcription. How does histone acetylation increases DNA transcription? Removal of histones positive charge. Removal of histone positive charge. What is the role of the H1 type of histone in chromatin structure? H1 histone, it binds the nucleosome and linker DNA to stabilize tighten chromatin structure. It binds nucleosome. Nucleosome binds the nucleosome.